This recording's for Biology 1407. This is a lecture presentation on material from Chapter 28 and 29 of the OpenStax textbook, the last block of material for Unit 2, Evolution of Animals, leading through to humans in the mammalian lineage. So if we take a look at the study guide, and let me increase the size of this just a little bit. There we go. So I'll walk through the study guide first, and then we'll present this material visually, and then talk about the associated homework assignment. So Chapter 28 covers what are referred to as invertebrates. Animals are somewhat disingenuously divided into two major groups, invertebrates and vertebrates. Vertebrates being really just a particular specialized group of a larger pile of things called animals, all of the rest not being vertebrates but having other body plans, equally diverse, equally sophisticated, but different. All right, so as we look through Chapter 28, we just want to walk through the evolutionary innovations that led to the great diversification of animals that we have. And we already covered this pretty substantially in Chapter 27, but this is meant to reinforce that material. So the first thing you want to start with is the ancestor of all animals, and we recognize that this is some sort of protistan animal, which simply means a single-celled animal-like eukaryotic cell. All right, so single-celled, not multicellular. Animal-like in the sense that it is a heterotroph and typically not covered by a cell wall of any sort, although there's a lot of variations and exceptions to that rule in the world of protists. It's a very complex group of organisms. All right, and we recognize that most likely ancestral animal-like protist would have been a flagellated single-celled organism that these days we would call a choanocyte, a single-celled filter-feeding cell existing either singly or in a colonial form, a bunch of them attached to a common structure. And that is something that we still see today, and they seem to be the most likely ancestors of all modern animals. The next major level of organization is the appearance of the multicellular state. All right. Now, that kind of started off slowly, so we've got a group of animals that are multicellular, but don't really have to be multicellular. That's kind of hard to explain, but when we look at poriferans, so sponges and all of their relations, and placozoans, what we see is, a multicellular organism that's made out of a whole bunch of different cells, including specialized cell types. And poriferans have structural cells, they have feeding cells, they have even a kind of immune cell. They're not true proper multicellular animals, only in the sense that they don't have true tissues. And if you break them apart into pieces, the individual cells still function. In fact, you can grind up sponges. Not all the cells will live, but eventually you'll get more sponges out of the debris as long as there's still living cells mixed in there. It'll take some time, but they're able to regenerate themselves in that sense. Placozoans are a very, very unusual little group of very small, disc-shaped or oblong shaped masses of cells, kind of like a jellyfish without any details, just a jelly blob, if you will. And they're a few layers of cells thick. They don't have any specialized tissues. They've got variations of cells, feeding and moving cells and all of this. And we find them in marine environments, very, very small, but a very early animal-like thing made out of lots and lots of cells working together. the appearance of tissues is the first major differentiation that distinguishes all other animals from these protist ancestors and from these peripheran sponge and placozoan relatives, and that is the development of true specialized tissues. All right, and the first organism or group of organisms that we identify as having those true tissues are the cnidarians, the jellyfish and all of their relations, and the ctenophorans, which are the comb jellies. The big difference between these is the cnidarians have a gastrovascular cavity, one way in, one way out, while the ctenophorans have a complete digestive tract. So morphological work puts them as being fairly closely related to each other. Molecular work actually puts them fairly distantly related to each other. So it's difficult still resolving the relationships between cnidarians and ctenophorans. But for our purposes, we'll just identify the fact that they have true tissues. They have diploblastic anatomy, which means they just have two layers of tissues, the endoderm and the ectoderm. There is no mesoderm, but instead there is a I can't think of the word I'm looking for, but a intermediate layer of jelly-like material that has structural proteins in it and occupies space, but it does not contain living cells. And that's what makes a diploblastic organism. Only those two layers of embryonic tissue, and indeed only two layers of adult tissue. They also have radial symmetry, and probably the ancestors of all of these guys had radial symmetry. It is, again, difficult to know precisely. But that's the first innovation, is the appearance of tissues, and we start with two tissues. All right. The next group of distinguishing features that distinguishes all other animals from all of these previous animals right here, the next evolutionary innovation, is the development of a triploblastic anatomy, where we have ectoderm, mesoderm, and endoderm. All right, and at the same time, we'll see that those organisms will have consistently a primary bilateral symmetry. So that means a left and a right, and a front and a back, and a top and a bottom. All right. Now, all other animals will be triploblastic and have that bilateral symmetry. All right. And we'll go ahead and put our platyhelminths in this particular location. These are flatworms that have a gastrovascular cavity, so they have a one-way in, one-way out digestive system, but they're triploid. They've got not triploid, triploblastic and bilateral. All right. The next innovation, and there is uncertainty about this, but we'll go ahead and call it the next innovation right here. But the exact sequence of evolutionary events that led to body cavities is still under study, so there's more than one way to present this information. But we'll go ahead and do it simply here and bring this in as the next major innovation, the appearance of a body cavity called a coela. All right, a body cavity is a coela. And animals that possess body cavities are often referred to as coelomates. Coelomates, animals with a coelom. And the coelom is this body cavity that allows the internal organs to develop and move around inside a cavity. It doesn't mean they're free-floating in there. They're still attached and plugged into all the rest of the body systems. So if you think of your own anatomy, think of your digestive organs in your abdominal cavity. That's a coela. Your respiratory and your lungs and your heart, basically, in your thoracic cavity. Your brain and spinal cord in your posterior cavity, the cranial and spinal cavity. And then you also have body cavities where each of your large joints end. Think of your hip joint, your elbow joint, your wrist joints, your finger joints. There's a tiny little cavity between each of those pairs of bones and those very flexible joints, and that is technically a body cavity as well. There's a fluid-filled space there, and it allows the bones to slide past each other without being fixed directly to each other. And that's the nature of a body cavity. It allows for much more complicated anatomy. And so the body cavity will be the next major innovation, and all other animals are going to have those body cavities. All right. there. Then we go ahead and take a look at those animals that have body cavities, and they are broken into three major groups. We have the Lophotrichozoans, these animals that have a lophophore, which is a filter feeding structure, all right, or a trochozoan larva, or both. And so there's a bunch of reasons that these animals are all grouped together. The ones that you need to pay attention to are the annelids, the mollusks, the brachiopods, ectoprocs, and rotifers. And each of those groups of animals is illustrated in the previous video. Annelids being our earthworms and their marine relatives, which typically have leg-like structures attached to them, not for walking, but for breathing primarily and scraping through the substrate. And also leeches. Those are all annelids. So earthworms, marine worms, and leeches. Mollusks are going to include clams, mussels, oysters, all of those guys, as well as snails and slugs, and also octopuses, nautiloids, and squids. So a very diverse group of animals with some pretty extraordinary members in them. The brachiopods on the surface, at first glance, look kind of like a clam or a mussel, but attached to a stalk typically, and that's then attached to a substrate. Unlike the mussels and the clams, though, instead of having an upper shell and a lower shell, they have left and right shells. They are symmetrical left and right, with a left and a right shell. And then when they open up, feeding legs that look a lot like an insect's legs stick out of the and that's why they're called brachio. That means arms right here. And they have these feeding arms that reach out from their shells. So it's a very different group of animals. Ectoprocs are small little sponge-like animals that are encrusting animals that live on the surfaces of rocks and make sheets of animals across the surface. They can be confused for a soft form of coral, and that is vaguely what they look like. Rotifers are very, very small filter-feeding animals that attach to a surface but then can move around freely in the water, and are named after their rotating feeding appendages surrounding their mouth. Another major group of protostomus, and I forgot to mention this, these are all the protostomes now. We'll divide the animals next into protostomes or deuterostomes. And those protostomes divide up into the Lophotrichozoans, and here's this group. We're going to just have them as a polytomy. We're not going to attempt to resolve the final relationships between annelids, mollusks, brachiopods, and ectoprocs, and rotifers in this course, although there are hypotheses about how they're related to each other. Another group of protostomous animals is the ecdysozoans, which refers to animals that lose their outer shell in order to grow, or their outer skeleton, and that is a chitinous outer skeleton. And there are two groups there, the nematodes, which have an unjointed outer skeleton. They look like worms. They are often called worms, but they are not worms in the same sense that annelids are worms. It's another worm-shaped animal. So the word worm really just covers a shape. It's not very specific, and nematodes are one of this groups. And there are free-living and parasitic worms. When you think of intestinal worms, that's what you're thinking of. It's a nematode. The bulk are free-living, but of course there are some parasites that we're quite familiar with. The other major group of ecdysozoans that you need to know about are the arthropods, a very large, diverse group of animals that includes insects, spiders, scorpions, and all of their relationships, horseshoe crabs, shrimp, lobsters, and all of their marine arthropod relatives, copepods, Daphnia that you worked with in laboratory. Those are all arthropods, and they have that external skeleton that has joints in it, arthropod. So jointed foot is what this name actually means right here. And there they are. And so those are the major groups of animals with body cavities that are divided up into two major groups. The protostomes and the deuterostomes, and the protostomes themselves are further divided into two major groups, the Lophotrichozoans and the ecdysozoans right here, the nematodes and the arthropods. All right. Then we move on, and so that's what you've got to be able to talk about. And let's let's stop and take a look at a phylogenetic tree to kind of get those in order. So here we'll take a look at a summary of that information, and this is a horizontally arranged cladogram showing the phylogenetic relationship between all of these organisms. So a cladogram, again, is just a branching diagram. When it's phylogenetic, that means it's based on evolutionary, hypothesized evolutionary relationships. So we'll start here with the metazoans, but if we go back in time, of course, we're going to have an ancestor that, all of these things were derived from, that all the metazoans come from, and that's going to be the choanoflagellates. So, this line leads from choanoflagellates, or we should say, down here would be a line going all the way to now, choanoflagellates, and then branching and having an evolutionary innovation, which is multicellularity, distinguishing everything on here from the choanoflagellates, which would be the next line down here. And we could even go back another step and say, the ancestral eukaryote, ancestor of all eukaryotes, one of which was, one of whose descendants was the choanoflagellates, and then one of the branches of the choanoflagellate lineage leads to all of this stuff. So the metazoans, again, that means, and generally this item, think of it here, the line continues on like this. The line continues on like this. The line continues on like this. Generally, the lower placement is considered to be the more ancestral state in this diagram right here. Not exclusively, but that's a good way to organize it. So the metazoans all possess multiple cells, the simplest of which are simply, metazoans, or called parazoa because they don't have true tissues right here, are the sponges and all of their relationships. The next evolutionary innovation, so I'd make a little line right here and say there was an innovation right here, and that is the development of specialized tissues that distinguishes all other animals from the metazoans. And so that's the true metazoans, true multicellular animals, eumetazoa. All right? And the basic groups right there considered to be the ancestral state are the cnidarians and the ctenophorans. By the way, these are Greek words, ancient Greek. And these two consonant pairs right here, it's the second consonant you pronounce. That's why it's cnidarian, ctenophora. You're like, imagine you're saying C without saying C, and then you get the accent right. But anyway, that's the logic of how they're pronounced. Ctenophorans, cnidarians. When you see those double weird consonants at the beginning of a word, you're probably looking at something Greek. Anyway, the cnidarians have those... what did I say right here? Yeah, the cnidarians are the jellyfish, the coral animals, sea anemones. All of those things are cnidarians. And then the ctenophorans are a free-swimming, vaguely similar looking, but actually structurally pretty different group of organisms that are called comb jellies right here. Both of them are radially symmetrical and have two tissue layers with that jelly-like material in between the inner and the outer layer. And that might be a vanishingly thin layer, or it might be a really thick layer, like when you think about a cannonball jelly from the coastline of Texas. Terrific amount of mesoglia in those particular animals. That's the name for the middle layer, mesoglia. All right. The next innovation is the development of triploblastic anatomy and bilateral anatomy, bilateral symmetry, and these seem to come together. At least there's no distinct, examples where one is true without the other. So, all current triploblastic organisms are bilaterally symmetrical and vice versa. And so that's the bilaterals. That's the name for this group right here. All the bilaterals include all of these things. So we are bilateral eumetazoans, right? But we are not radiata. And then we can divide the bilaterals. The ancestral state, is assumed to have had no body cavity. And so that's why we go ahead and sometimes put the platyhelminthes down here, sometimes not. So that's why this is an odd group out here. Some reconstructions put them very firmly in here with the Lophotrochozoans. Others put them outside as an ancestral acoelomate animal. There's some other phyla collectively referred to as the Acela. They're all marine. They're all relatively low in diversity, although there's very large numbers of these things, but there's not a huge amount of speciosity out there. But no body cavity. The rest have a body cavity, so those will be the coelomates as opposed to the acoelomates. So the coelomates all contain a body cavity. And there's really very little information as to which of these two comes first. So we've got a split right here, but we can assume one of these groups had to be the ancestral state, but there's no real way to assess that currently, at least that I'm aware of. So we'll just take the coelomates and divide them up into the protostomes. That is where the digestive cavity, as it works its way through the animal, the digestive cavity, the first opening is the mouth, and the second opening, when it develops somewhat later, is the anus. And so the digestive cavity simply runs from front to back in the early developing embryo. The deuterostomes have that reversed. The first opening will be the distal end of the digestive system, the anal opening, and then the last opening to form, still very early on in embryonic development, for us within, the first week or so. The second opening will appear in that early embryo, and that'll be the mouth, the deuterostome. So the mouth is the second thing. The mouth is the first thing in the protostomes. Mouth is the second thing in the deuterostomes. All right. And then we're going to take those protostomes and we're going to divide them up into two major groups. Again, there's no real polarity here as to which is the more ancestral group. There's arguments that can be made on either line, although generally this group will get thrown in as probably being representative of the more ancestral state. And that's the Lophotrochozoans, and the Lophophore is a ciliated feeding apparatus that sticks, out, that is associated with the mouth, with the oral opening, and it is a filter feeding structure. And a lot of these things have them as adults or as embryos, or some, in some developmental stage they have them. And then the trochozoan is a particular larval form. A larva is a lot of these things like brachiopods and ectoprocs are sessile, and a lot of mollusks are, like clams. They sit where, they live where they sit. But the young are free swimming and ciliated multicellular things that swim around in the ocean currents, mostly getting just washed around by currents. But that's the dispersing larva, and that's a trochozoan. Not all of them have them, but it's seen one way or another in most of these groups. The platyhelminths, again, are an odd group right here. One of the weird things is, even though they are here classified as a protostome, they only have one opening in their digestive cavity, so that kind of puts a problem in this protostomus versus deuterostomus anatomy right here, and which is why some of them are brought down here into the acoelomate group. So we'll just kind of put the platyhelminthes in that strange. I'll put them down here. Let's go ahead and call them acoelomates. And then that leaves the rotifers, the ectoprocs, brachiopods, annelids, and mollusks as our representative Lophotrochozoans. They're a lot easier to work with. And we won't resolve them any further than this. So we'll just keep this as a polytomy. That means the work is not done. There is a branching diagram here, but there's too many different hypotheses still being tested, data being collected to work out which is the correct one here. The ecdysozoans, the other major group, again, characterized by their shedding of their external exoskeleton. The nematode, we will assume, will be the ancestral state, and the arthropod with the jointed legs, the more derived version. So we could put another innovation right here, which is articulations, joints in the exoskeleton. And that would be the innovation that the arthropods have. All right. So that's a quick look at that material. Let's go back to, our study guide right here. What happened to this? Why did that? At 100% there. Okay. Okay. Now, the next part, we'll take a look at we want to take a look at the echinoderms, the echinoderms, and the chordates. So let's go back and take a look at this branching diagram here just for a little bit. So this is the deuterostomes. The deuterostomes divide up into the chordates and the echinoderms. And which came first right here, very hard to tell. The echinoderms are a highly modified group. These are starfish, sea urchins, sea cucumbers, sand dollars, and a few other groups, but they all have what's called five-fold secondary radial symmetry. So that means as an adult form, the juveniles are, the larvae are different oftentimes, but the adults have symmetries that are multiples of 5, including 5-armed starfish. And if you look at a sea cucumber, it's got 5 sides. If you look at a sand dollar, you'll notice, if you look carefully, it's got 5 lobes, or there's 5 rays making up its body. And same thing for sea urchins. Internally, their anatomy is very clearly left-right, though. So they do have a left-right internally, a top and a bottom, very obviously for all of them. And if they have a left-right, then they've got a front and a back, technically. So on a starfish, there's one arm that's facing forward, 2 left arms, a left and a right arm, and a left and a right rear arm, if you will. And they often even lead with that, lead arm. So there is directionality and specificity to them. So that radial symmetry is secondary. Internally, they are bilateral. All right? Deuterostome bilateral. And then the chordates is everybody that's got the first anatomical evidence of a backbone. The chordate itself is named after the notochord. It's a flexible bit of cartilage in the back of the animal, so not a true backbone, but it's the beginning of that lineage, and that's the chordates, the 2 major group of the deuterostomes. Now we'll go ahead and take a look at them. So if we go back to our study guide, you've got to be able to make those statements about the echinoderms. Oh, I forgot sea lilies as well, the crinoids, which are like starfish attached by a stalk, upside down starfish waving around, filter feeding in the sea, attached to a stalk attached to the ground, or to the bottom of the ocean, or rock typically. Anyway, very interesting things. Look them up. All right, so they're a very, very neat group of animals, but we won't spend much more time on them. Then we take a look at the chordates, and the chordates we're going to distinguish into 3 major groups. All right. And that is the tunicates right here, which are called sea squirts. It's a really weird looking little animal. The adult form attaches to a rock and is a filter feeder. It vaguely looks like a highly organized sponge. It looks like a very giant ectoproct, another encrusting animal. But it's more closely related to us than those other things, in the sense that it is a chordate. The adults don't have them, but they're juveniles. The juvenile form looks a lot like this other thing, which is the cephalochordate, called a lancet. I'll show you images of these things in just a little bit. The cephalochordate is something that if we looked at it, we'd go, that's kind of like a weird fish, a weird primitive fish. And the larval form of the tunicates looks just like that. But then as an adult, they turn into this strange blob-like thing. They lose their brain, they lose their notochord, they lose a lot of structures. The cephalochordates live as adults, in this tiny little primitive fish-like thing, but they don't have a lot of the features that we normally associate with chordates. And then the proper chordates are characterized by that stiff fibril skeletal element and all of these other things, and particularly a great big filter-feeding perforated structure. That is the beginning of the larynx, and it's this filter-feeding structure right here that is the origin of our larynx. It's the origin of gills. It's the origin of a whole bunch of anatomy and structure that's retained in all the rest of the chordates. All right. So the 3 groups are the tunicates, the cephalochordates, and then the true chordates, which are the. We'll take a look at those in just a second. All right. And the other thing is that the body is organized into these muscular groups over and over. They're called somites, these regular muscular groups, and then there'll be regular skeletal components associated with that regular musculature. All right, a really good example of that anatomy, if you look at a fish and look at the musculature of the fish, look at a snake, look at our thorax, just our torso. Not our arms or our legs, but our torso is divided up into these repeating regions, and that's that sort of anatomy right there. All right. There is a posterior tail that extends beyond the anus. We, of course, have lost this tail almost completely. We have a tiny little skeletal tail, but as a diagnostic feature, chordates have a tail that goes beyond the end of the digestive system, postanal tail. All right. The most specious and diverse group, as it says right here, of the cephalochordates, things that have a head, are the vertebrates. And, that's things that have here we've got a skull, and then we have a notochord, and then the vertebrates have an articulated notochord. The instead of a whip-like or a rod-shaped piece of cartilage, it's broken up into a piece of cartilage connected by soft, more flexible material, basically the intervertebral discs, and then another piece of cartilage, then disc, then cartilage, then disc, then cartilage. That's the vertebrates. So the oldest, the most primitive vertebrates, the most ancient vertebrates, and a whole bunch that are still around today, their bones are made out of cartilage, not hard mineralized bone. So the cephalochordates and the early vertebrates, including vertebrates that are still around today, think sharks, rays, and skates, and all of their friends, are all they have a cartilaginous skeleton. But they have this articulated skeleton and a complete skeletal structure surrounding the nerve cord. So the most primitive chordates, they've got a notochord, the stiffening rod, and then vertical to that or dorsal to that, they'll have the nerve cord, the brain and nerve cord, but none of that is protected. By the time we get to the cephalochordates, we have a capsule of cartilage surrounding the brain, and then in the vertebrates, that capsule of cartilage goes ahead and surrounds the nerve cord as well, and it's articulated so that it can bend, and that's the vertebrate skeleton. All right. The most primitive group, and they're still around today, and these would be the ancestors, are agnathans. All right. So this is a jawless fish that's got a rasping sort of open mouth with little tiny teeth around it. The first mineralized structures are teeth for gnawing holes in the sides of their prey, whatever that may be. And these things are still around today. We have them. They're called lampreys. There's a type of fish called a lamprey, which is a parasitic fish. It's an ectoparasite, like a tick, that attaches to the outside of its host. And it sucks fluid, blood and, body fluids out and rasps a hole in their body to do that with their open, jawless mouth. Very, very distinct looking animal. The next major innovation is the development of a jaw from the first of the rib arches, and that is the gnathostomes right here. So gnathostomes are jawed cephalochordates. So they've got a skull, or they're jawed vertebrates, if you will. And what's happened is the very first two arches of the gill arches, the highly modified larynx, I'll show you imagery of all of this in a moment, has been modified to make a jaw. And so the jaw allows for more aggressive predatory feeding than the agnathans are capable of. Typically, because they're chasing after things with their jaws, they've got improvements to their swimming abilities in the form of additional sets of fins, which make them able to swim and chase after prey more effectively with their jaws. And some really fabulous, huge ancient fish came into being as a example of these gnathosomes. An entire extinct lineage of bony jawed fish are the placoderms, which are fish with external armor. There are armored fish today, but they're not part of the same lineage. These have all gone completely extinct. But once upon a time, they were a very impressive group. And what they had was a cartilaginous skeleton and then a bony, hard armor on the outside of their body. All right. Now, the others, there are, some gnathosomes also went in another direction, and a big evolutionary innovation in them is are two, the chondrichthyes and the osteichthyes. So the chondrichthyes might be the more primitive group, and that is modern fish that still retain that cartilaginous skeleton. The skull, the vertebral column, the ribs, everything are made out of cartilage. And that's, of course, sharks, skates, and rays. If you're not familiar with skates, they're sometimes freshwater. They're also freshwater rays, but smaller, a little bit different than the rays, manta rays, and sea rays, and all of those guys. And, of course, sharks everybody knows about. Collectively, they're referred to as chondrichthyes, and indeed the word chondro refers to cartilage. Chondro, we'll see that in human anatomy. Everything having to do with your cartilage, the proper terminology is chondroitin. You'll see that nutrient you can buy in health food stores is chondroitin, which is a chemical component of cartilage. The other group is the osteichthyes, which are the bony fish. And here what's happened is, that the hard mineralized stuff that the agnathans first showed up with in their scraping jawless mouths And excuse me, and the placoderms took to great use to make these plates covering the forward part of their body, including great big slicing bony jaws, if you will. The Osteichthyon fish used that same set of genetic tools not to create just the teeth and not to create the external armor, but instead to mineralize the cartilaginous skeleton. And that's true for all all the rest of the vertebrates, is that they have a mineralized skeleton. If you're not familiar with it, when you start developing as an embryo, you have a purely cartilage-based skeleton, and then as you continue developing, slowly portions of that cartilage-based skeleton are converted into bony skeleton. Very, very clear that, our phylogenetic relationship to rays and sharks and all of that is recapitulated in our development. We go through a cartilaginous phase before developing our bony skeleton as adults, or as, maturing our way towards adults. All right. In the world of bony fish, one major group that distinguishes itself are the Sarcopterygiids, fancy sounding name, and this is a group of bony fish that have pairs of really, really stout fins. Two pairs, a pectoral pair, and then a pelvic pair. And if you're, thinking about those terms, that refers to arms and legs. So they've got a pair of muscular fins where our arms would be, and they've got a pair of muscular fins where our legs would be on our body. Right about even with the end of the digestive system. And those are the precursors of the forelimbs right here. So the Sarcopterygians are these bony, fleshy, lobe-finned fish. They're oftentimes referred to as lobe-finned fish. And what they did is they developed in freshwater, probably, shallow freshwater habitats, and dragged themselves across, periodically exposed mudflats. If you're aware of it, all freshwater habitats, rivers anyway, especially the bits close to the ocean, tend to rise and fall due to tides, in the estuaries. So, these muddy habitats are a big, big deal. They were a great habitat for getting food, but it created some, major moving problems for large fish-based organisms. So that's kind of where the Sarcopterygians come from. Tetrapods are an innovation. This is a group of Sarcopterygians whose limbs became much more robust and allowed them to actually move onto land and move around on land. And we would call these things amphibians. The tetrapods, the earliest tetrapods are the first amphibians, which is simply a class of fish-like animals that can move around on land, have adaptations that make it capable for them to move around on land, breathe air for an extended period of time. But if you're familiar with your amphibians, frogs, think more, less frog, less toad, think more salamander or newt, four limbs and a long tail and a more reptile sort of body shape. Well, that's the earliest tetrapods. All right. Frogs and toads are highly, highly specialized, relatively recent, relatively recent, last couple of 100 million years, innovations in that lineage. All right, so they would have been like wet reptiles that lay wet eggs in the water, and we know that that's the earliest tetrapods, also called amphibians. The next major innovation leading to us is that some terrestrial amphibians, terrestrial tetrapods, developed a waterproof egg, and that's the amniotic egg, surrounded by a very, very tough fibrous membrane that allows for the exchange of gases but retains water. And that's a terrestrial egg. So there's a group of these terrestrial tetrapods, which are in fact ray-finned fish, which are in fact bony fish, but adapted for living on land, that developed a waterproof egg that they could lay on land. And that, of course, is going to be the first of what we call reptiles these days. All right. Now, in the reptile lineage, there's a major diversification way back in time. Between diapsids and synapsids. All right, so synapsids is a group of early reptiles that had one opening in or depression in their skull. It's an attachment point for musculature having to do with the jaws. And these guys turn out, and they're a very early group of reptiles. They tend to they diversified really, really terrifically. Almost all of them went extinct 250 million years ago, but one lineage made it through, and it's the ancestor of all modern mammals. So we are a synapsid reptile. All right. The other big group, the diapsids, having two of these depressions on either side of the skull, is the great diversity of reptiles we normally think about, and that includes the crocodilians, everything that we call a lizard today, and of course the group that diversified from the crocodilians, the dinosaurs and their descendants that are still around today, the birds. And those are all diapsid reptiles, and they actually came as a big diverse group after the synapsids. Synapsids got off to an early start, what are called the mammal-like reptiles. And again, their time when they were really, really doing well on Earth was in excess of 240 million years ago. At the end of that point in time, there was a major extinction event. And most of the mammal-like reptiles, which were really well distributed across the Earth, very, very large animals, very large numbers of them, died off, with the exception of a couple of their smaller the smaller groups, and at least one of those made it all the way through to the present. And that would have been, what were at the time mammal-like reptiles that would have looked like little rats, sort of rat-like things, and they made it all the way through to now. And of course the mammals have diversified substantially in that time. The diapsids were extraordinarily diverse and well distributed all over Earth, but again, most of them have died out with the exception of large numbers of lizards, crocodilians, and birds. The synapsid reptiles that diversified into a new group are the mammals, the true mammals, and what distinguishes them from their synapsid ancestors is the retention of a glandular skin, which is what the terrestrial tetrapods, these guys produced a mucus-like, slime that covered the outside of their body, and the amniotes dispensed with that, most of them, as they adapted for life more on land by keeping themselves from drying out as much. But some of the groups retained a little bit of a glandular skin. Others had a much more heavily armored skin, think, reptile, not exoskeleton, but, the external scales of a reptile. But some of them were not protected in that same sense, and so they retained some glandular characteristics to their skin and ultimately produce hair, cover that skin with hair. And the glandular component is the musk, the smell glands, because lizards don't make musk, but mammals do. To mark territory, of course, milk and sweat. Not all mammals sweat. All mammals make milk, and most make some sort of odorous fluids that they release to mark territory or communicate in some way. Another big deal with mammals, all modern mammals, and their ancestors, including the synapsid ancestors, is that they had diverse teeth for different jobs in different parts of their head, whereas the diapsids all have the same sorts of teeth in all places in their heads, or indeed lose their teeth altogether. And then thermal control, homeothermy, which was also developed in the diapsids. So this is probably, the homeothermy probably came before the split between synapsids and diapsids, but it's very hard to tell. It either showed up twice or it was lost secondarily in a bunch of groups. But anyway, that's the mammals. Then, as we're working our way through, in the mammals, we want to look at three major groups, and those are the monotreme mammals, which we would consider to be most like the ancestral modern mammals. The platypus and the echidna are the examples. And what makes these guys unique is that they still lay eggs. It's an amniotic egg. It's a waterproof egg, but they do lay eggs, just like a bird does, or like a snake does, or a lizard does. And their milk glands are not organized into breasts. They just have lots and lots of milk glands on the underside of their body. They just sweat milk, if you will, and the young lick that up. A group we're much more familiar with is the therian mammals, which means these animals, the egg hatches internally inside the womb. And the most primitive group of these are the marsupials, where, that very, very, that they retain eggs. The eggs are kept inside the body. The egg hatches inside the body, and, they give birth to a live young. In the marsupials, it's very, very underdeveloped, crawls into a pouch. They have this pouch structure. That's what a marsupium literally means. It's Latin for pouch. And the embryo, really, attaches to a mammary gland there, a breast, and is attached to it, fixed to it for several months as it grows and develops in the pouch. What's interesting about the marsupials and why they do really well is that they can be pregnant with a developing embryo, one or two of them in the uterus. A very young or even two young individuals attached to the two breasts inside the pouch, and an older individual from a previous generation that's still living in the pouch but able to hop out and go around and feed with its mother and then move back into the marsupium. So they can be carrying up to 3 generations of young at the same time, which is a pretty good reproductive strategy. The other major group of mammals is the placental mammals, and of course we are placental mammals, and what happens there is the egg has an amnion, but it's also plugged into the uterus via a placenta. All right, so that amniotic egg is retained internally and attached to the mother by a placenta, and the embryo grows to great size, much more than the yolk of the egg would allow, because we have a yolk in us, but it's very tiny. We don't have a big yolk because we don't need it, because we're going to be plugged into a placenta. So, placental mammals still have yolk-filled eggs, but they're tiny, tiny, tiny eggs with a very small amount of yolk. And the placenta is the major innovation right there. That gives rise to all of the mammals. We'd love to study all of mammal diversity, but we can't in this particular case. We're just going to take a look at the major divisions in mammals that will give rise to humans right here, and that is the primates. The primates are one of the major groups of mammals that's distinguished, as it says here, by binocular color vision. So binocular vision, meaning both of our eyes look forward. We're not the only binocular vision mammals. The carnivores are they all have really good binocular vision. Bats, chiropterans, also do. And, to various degrees, all mammals are somewhat binocular, but they don't have forward-facing eyes. They usually have left-right eyes and see a much bigger world around them, but only a small portion is binocular. Anyway, a lot about vision. The other big one, and not totally unique to the primates, but a characteristic of them, is particularly good full spectrum vision. It doesn't mean sharp vision. They can't see really far necessarily, all of them, but they've got great color vision. And in our cases, we've got pretty good long-range vision as well as good color vision. And then the thing that makes primates to most of us is the opposable thumb, which is a digit that can reach across the other digits and oppose any of the other digits. And that is a unique thing. There's a number of other animals out there that have grasping claws, but they can't oppose their digits to each other. So raccoons come close. Procyonids, a group of mammals that raccoons are, but they can't do this. They can't hold something between 2 digits under pressure in the same way that we can, because they don't have that opposable digit. All right? And a lot of primates, the big toe is opposable just in the same sense. On us, it's lost because our foot has been adapted for exclusively for walking and not very good for climbing anymore. But our ancestors certainly had an opposable toe as well. That's the primates. And then if we take a look within the primates, we're going to have the Platyrrhinians, which is a group of primates that includes all what's called Old World monkeys and apes. The distinguishing characteristics of the Platyrrhinians, or the Old World monkeys, is the anatomy of their nose. They have a flat, sort of opened up nose. Some of the hominids have changed that, but it's a characteristic there. Also, the Old World monkeys, they have a tail that is non-prehensile, right? The Platyrrhinian apes that have lost their tails. The Platyrrhinian primates, are, as I said, monkeys right here. They have a tail that they use for balance, but it is not a grasping tail. If you think about that, if you've heard of, or you see pictures of monkeys hanging from their tail, they are what are called New World monkeys, or I forget the proper name. It's not Platyrrhinian, it's another name for the nose. Rhinon means nose. Anyway, they have prehensile tails, and they can use that as an extra limb, if you will, whereas the Old World ones cannot. All right? Big innovations are terrifically complex social structure, opposable thumb, etc., etc. The apes are a derived group of old world primates that have lost their tails, so they don't have that postanal tail any more than we do. They've got a little coxal bone, so there's a tiny, tiny embryonic tail in there, but all the external tail is lost normally. Brains typically are quite a bit larger. They exhibit, collectively more complex behavior. The great apes are going to include gibbons, orangutans, gorillas, chimpanzees, bonobos, which is another species of chimp, and humans and all of our extinct relatives. The hominids, finally, is a group of derived ape primates that are specialized for terrestrial life rather than brachiating, so they walk around on the ground. They're upright, bipedal, substantially enlarged brains, sophisticated social structures. We are hominid apes. Australopithecines is the first major group, Paranthropus another major group. The Australopithecines, as distinct species, have all gone extinct, but at least one of their lineages leads all the way to the lineage of Homo. The paranthropal apes all died out. The Homo lineage diversified into a number of species: Homo neanderthalis, Homo denisovian, and Homo sapiens. The Neanderthals and the Denisovians have gone extinct, but the Homo sapiens are still around, and of course that is us today. Let's go ahead and take a look at some phylogenetic trees to illustrate the sequence of events. So if we look at fish, the agnathans right here, there's the lampreys and the hagfish. I forgot to mention them, a really wonderful group. So these are vertebrates. They are cephalochordates that are jawless. That's the agnathans. So cephalochordates would be the term we would put up here, cephalochordates. The next group, the big innovation, is the development of a jaw. That's the gnathostome. So everybody else, the cartilaginous fish and on down, are all gnathostomes because they've got this jaw made from the first rib arches. And assumably the most ancient, the most primitive, the oldest lineage, and indeed the fossil record pretty well supports this, are going to be the cartilaginous fish. That's the sharks, rays, and skates and all of their relatives, the most ancient gnathostomans. The osteichthyans, the innovation right here is the bony skeleton. So that's the ray-finned fish right here. That's the great bulk of fish that we think of out there. And then we also have these lobe-finned fish, right? The sarcopterygians, which is the development of the lobe-like limb. And a bunch of these are still around today. They're a lungfish and they're a coelacanth. Coelacanths are deep water fish these days. They're freshwater, shallow water relatives have all gone extinct or have changed so much we don't recognize them. And indeed, that's what these guys all are. Lungfish are still around today and inhabit freshwater, shallow, irregular waters, which is why they have this lung, this breathing lung. Anyway, that's the osteichthyans, and of course a major group of the osteichthyans, that's the sarcopterygians, the lobe-finned fish and all of their relatives. A major group of sarcopterygians really specialized their lobe fins. That's the tetrapods. So this includes everything that we call a amphibian today. Salamanders are the most, probably most like the ancient tetrapods that first moved onto land. Again, frogs and toads are highly specialized. And the caecilians are a limbless amphibian. They've secondarily lost their limbs, and they kind of, in moist soils, they kind of like snakes have lost their limbs. There's lizards and snakes. Snakes lost their limbs secondarily from a lizard ancestor. Caecilians lost their legs secondarily from a salamander-like ancestor. A big innovation in the world of tetrapods. These guys did really great, but they're always needing to go to water to reproduce, mostly one way or another, is the amniotic egg. So the amniotes, develop this fabulous terrestrial egg that allowed them to move away from water and indeed, really cut their dependence on water down to the bare biological minimum needed to stay alive. And we'll divide those, if not on this chart right here, but into the diapsid amniotes. That's going to be dinosaurs, crocodilians, snakes, birds, everybody. And the synapsid amniotes, which is the ones with the one, dent behind their eyes, for muscle attachments, and that leads to mammals. And we've got the monotremes, the egg-laying mammals, the marsupial mammals, and the placental mammals. All right. The last thing we want to take a look at is the hominids. So let's take a look at this. I'll see if I can make it a little bit bigger. Okay, let's see if this works. Yeah, I'm trying to make this a little larger. And, here's the relationship. You don't need to know all of these terms, just the ones on the study guide as far as testing purposes. But here's the relationship between tailed apes right here. So here's a baboon. It's got a tail. That's going to be the platyrrhinid the platyrrhinid primates. So tailed old world monkeys, there's your representative of it right there, the baboon. And then the hominoidians are the the apes, and that includes gibbons, orangutans, gorillas, chimpanzees, etc. And then we break the hominidae away from the hominoideae. The hominids are the ones that are most human-like, hominid right here, and so that includes the gorillas, chimpanzees, humans, and all of their extinct relatives, the hominids, right? And then the homini is strictly the human lineage leading all the way to modern humans now. And those are the bits that you need to know. Just hominoidea, hominidae, hominini, that's kind of a tough one to say, hominini, and finally the humans, Homo sapiens themselves. And that is the recorded lecture on Chapter 28 material. Sorry, Chapter 29 material, 28 and 29.